Well, hello, I'm Mark McClellan. I'm the director of the Duke Margolis Center for Health Policy. I'm very pleased to welcome all of you to our webinar today on endpoint considerations to facilitate drug development for Niemann-Pick Type C, key themes and future directions from the January 22 public workshop. This is being convened by Duke Margolis in collaboration with our partners at the FDA. The webinar is intended to be a concise follow-up to the January 22 workshop on NPC endpoint considerations. And throughout the webinar, our speakers are going to provide some reflections and some distillations of the key themes and future directions that were shared during that January workshop and presented in the previously released workshop summary report and addendum. We hope that this event is going to serve as another touch point, another opportunity for a step forward in NPC drug development for stakeholders and FDA working together on this important topic. As you all, or many of you I know, are already too well aware, Niemann-Pick Type C, or NPC, is a rare genetic disease that typically results in progressive neurologic symptoms and organ dysfunction. NPC has historically been considered a childhood onset disease, but an increasing number of patients with NPC are displaying symptoms and onset later in life. Currently, there are no therapies approved in the United States for treatment of NPC. So in order to advance NPC drug development, it's important for stakeholders to work together to identify strategies to support both ongoing and future NPC clinical trials and evidence development. Indeed, Congress recently recognized the importance of this collaborative work. And we appreciate the FDA's efforts to support this collaboration, and very much appreciate all of you who are joining today to continue the work towards this goal of more effective therapies. As with the January workshop, the content of this event will focus on endpoints for NPC clinical studies generally. Specific drugs or use of expanded access are not within the scope of the event or the follow-up discussion today. And so as you'll see in the agenda, we're focusing on further challenges in impacting NPC therapeutic development and patient care. Ultimately, patients and their families are at the heart of this important issue. And as with the January workshop, we've incorporated perspectives from the patient community into the event to keep the focus on what's meaningful for patients and families. Before we begin, just a few additional housekeeping notes. I think on the next slide, just a reminder of the statement of independence at Duke Margolis. We don't take partisan positions, but we encourage everyone else to be independent in their views and to state their views clearly. More about our policies on independence, our links to more information is provided on this slide. And just an additional note that we are convening today's event in cooperation with the FDA. And another way of saying what I just said, the views expressed in the written workshop materials and the publications and by the speakers and moderators do not necessarily reflect official policies of the Department of Health and Human Services. And a final reminder that I'm an independent board member at Alignment Healthcare, Cigna, and Johnson & Johnson. And with that, I'd like to turn on the next slide to the agenda for today. We've got a pretty packed agenda. We're joined by a group of subject matter experts who are going to provide some remarks during this session, as well as a number of stakeholders who have registered for the public webinar. Again, we're very glad to have all of you with us. The meeting materials are available on the Duke Margolis website. We do want to hear from everyone here. If you note on your Zoom window, you should see a Q&A tab probably at the bottom of that window. You can put a question or comment in there anytime. Our participants may be able to respond to some during the course of the meeting. And then as you see on the agenda, we've got a bit of time set aside towards the end of the hour so we can keep everyone on time for bringing up some of those discussions, some of those questions for further discussion. A recording of this meeting as well as the slide deck are going to be available on the Duke Margolis event website and YouTube channel after the conclusion of this event. So with that, I'd like to get us started by turning to Dr. Patrizia Cavazzoni for some opening remarks from the FDA. As many of you know, Dr. Cavazzoni is the director of the Center for Drug Evaluation and Research at FDA. Patrizia?
Thank you, Mark. I am delighted to be here to kick off this very important workshop as a follow-up to the earlier workshop that took place this year. As we all know, there are currently no approved therapies in the United States for the treatment of Niemann-Pick C disease. And endpoint considerations are critical in facilitating drug development for NPC. The successful development of new treatments for rare diseases such as NPC is a key priority for us at FDA. In rare disease drug development, including NPC drug development, there are many challenges. And some of them include the fact that NPC clinical trials can be challenging given the heterogeneous manifestations that can be slowly and variably progressive. And we see this with a number of neurodegenerative diseases, even ultra-rare ones. As NPC is a highly heterogeneous disease, the clinical endpoints that are selected may not reflect disease presentation or clinically meaningful improvements for all NPC patients. In addition, there are only a limited number of patients with NPC who are available to participate in clinical trials, and this may restrict study design options. So at FDA, we see these challenges in the development programs we oversee, and we want to facilitate the development of NPC treatments and want to collaborate with the NPC stakeholders and the NPC community. Now, although progress has been made in drug development for a number of rare diseases, significant work still needs to be done, including work for NPC and to advance treatments for this terrible disease. So as we think broadly about the challenges in rare disease product development, it is really essential that we continue to expand our scientific understanding of rare diseases and build the tools needed for drug development. This includes, but is not limited to, new biomarkers, in vitro models, in silico models, clinical outcome assessment measures, innovative trial designs, and other tools where multi-stakeholder collaborations will likely be critical for success. Now, a key aspect of today's webinars is thinking about how we can leverage the important information shared at the January 22nd workshop to think about next steps to facilitate NPC drug development. There are really three initiatives that I would like to highlight to show you how we can continue to facilitate rare disease drug development. The first one is in May 2022, CDER announced the launch of a new Accelerating Rare Disease Cures, or ARC program. Now, the vision for this program is to speed and increase the development of effective and safe treatment options to address unmet medical needs of patients with rare diseases. And so through scientific and regulatory innovation and engagement, CDER's ARC program will support the development and approval of safe and effective rare disease treatment options. So in our first year, CDER's ARC program will focus on strengthening internal and external partnership with stakeholders and in engaging with external experts to help identify solutions for challenges in rare disease drug development. And one example of this would be a conference that took place on May 22nd, sponsored by FDA, CDER, as well as NCATS at NIH, in focusing on regulatory fitness in rare disease clinical trials, with still available in this, the proceedings of this workshop are still available online as a learning resources for stakeholders in rare disease drug development. The second initiative is to support innovation and quality in the drug development pipeline for rare diseases. And to that effect, FDA is also funding the Critical Path Institute work in collaboration with the national organizations for rare diseases to develop a rare disease cures accelerator data analytic platforms. This is an initiative that provides a centralized and standardized infrastructure to support and accelerate rare disease characterization with the goal of accelerating therapy development across rare diseases. And third, we are working with the Critical Path Institute and plan to provide grant funding to explore the feasibility of launching a consortium effort focused on advancing drug development in lysosomal diseases. We anticipate that this effort would complement the Rare Disease Cures Accelerator data analytic platform, as well as other rare disease collaborative efforts at Critical Path. And so to conclude, these collaborative efforts will help support the goal that we share with the NPC community to bring effective and safe treatments to patients as efficiently as possible. There are challenges, and we really want to work with the community, the stakeholders, and the scientific experts to identify those challenges and overcome them so that we can accelerate the delivery of treatments for these terrible diseases. So thank you for having me today, and I look forward to today's discussion.
Thanks very much, Patricia, for those opening comments and for those updates on some of the new activities underway that FDA is supporting. Next, I'd like to turn to Dr. Justin Hopkin for our first patient community perspective on the key themes and future directions shared at the January workshop. Dr. Hopkin will be providing remarks on behalf of the National Niemann-Pick Disease Foundation. Dr. Hopkin?
Thanks, Dr. McClellan, and thanks to Dr. Cavazzoni for kicking off the meeting, but I think highlighting three key initiatives that we are also very interested in and currently exploring in the NPC space. I appreciate having the patient voice being incorporated in today's opening remarks. I had the privilege of providing closing remarks in our first FDA listening session in April of 2021. And at that time, I outlined the following three take-home points on behalf of the community. The first point: please use regulatory flexibility when considering how we design clinical trials for NPC. The pitfalls of traditional clinical trial designs are magnified through the heterogeneity and variable clinical progression of NPC. Please partner with us to design novel clinical trials and with in-person analysis to limit the role of placebo by using our valuable natural history data. The second point I made: when clinical trial results demonstrate safety of a drug but may not reach the predetermined primary endpoints, please use your regulatory flexibility to accept real-life data that may suggest that a drug is effective over a longer period of time or in subgroups of patients. And the third point: multiple effective therapies will be needed for NPC. There are no cures in the pipeline, and our community's goal is to render NPC a chronic disease, and this will require multiple approved therapies, as we see in other chronic multi-system disorders. In our community, we have experience with 4 agents in our pipeline that have had a positive risk-benefit profile based on community and real-life experience. Now, I realize these points don't necessarily directly pertain to our topic of endpoint discussion, but we can't lose sight of what our primary goal should be today: supporting our current NPC patient population, meeting their immediate need for effective therapies, and valuing their experience and their data generated through clinical trials and expanded access programs. As has been stated already, NPC is a rare, progressive, universally lethal condition with no approved disease-modifying therapies. We've brought multiple therapies forward for evaluation to the FDA, but have yet to get an approval despite community backing with favorable perceived risk-benefit profiles. Our community has a high tolerance for risk as well as uncertain benefit. We implore the FDA to use its regulatory flexibility to its full extent when considering each of our current therapies in the pipeline for approval. The downside to this approach is minimal because we know the outcome of untreated NPC. Please don't let perfect be the enemy of good right now. Please help our patients today. In regard to the clinical endpoints in the discussion today, I would like to thank the FDA representatives here today, including Dr. Cavazzoni, Dr. Maynard, Dr. Karp, Dr. Noble, Dr. Donahue, and really the agency as a whole for engaging our community. We've had a PFDD, 2 listening sessions, a Duke Margolis workshop, and now this follow-up webinar. So we know you're listening. We appreciate your continued engagement, and we look forward to discussing and identifying a clear path forward today. A heartfelt thanks to Dr. McClellan and his team for productive listening session and a thorough summary that captured the conversation and salient points each speaker conveyed in January. In reviewing the summary document from our Duke Margolis workshop, our community feels that we were heard, and we hope to leave this meeting with a clear roadmap that will allow industry regulators in the NPC community to agree on validated endpoints that are meaningful to the community and reasonable based on reasonable clinical trial design. Last, we want to express our sincere gratitude to our outstanding scientific and clinical leaders for their untiring advocacy, outstanding compassion, and tremendous work in our space. And I would like to thank the NPC community for their advocacy, their perseverance, and unwavering commitment to each other. Thank you.
Justin, thanks very much for your thoughtful remarks. And now I'd like to turn to Dr. Sean Kaysen for our second patient community perspective on the key themes and future directions shared at the January workshop. Dr. Kaysen is the director of the Araparsigan Medical Research Fund at the University of Notre Dame. Dr. Kaysen?
Sure. Thank you. So my name is Sean Kaysen, and I'm the director of the Araparsigan Medical Research Fund at Notre Dame, which was founded by Mike and Cindy Parsigan in 1994 when 3 of their 4 children were diagnosed with NPC disease. Since then, we've been dedicated to support research to find treatments and a cure. In 2019, we hosted a patient-focused drug development meeting to bring the patient voice and perspectives to government regulators and industry. At that time, we felt this was a big leap forward for our community as we had multiple active phase 3 clinical trials. But in all honesty, if you ask most members of our community, they feel things have gotten worse and often feel that regulators, companies, and patient groups are talking past each other without coming together to wrestle with the fundamental issues. And now none of these phase 3 therapies are approved. And in the past 18 months have been even more brutal as parents worry daily that they may lose access to multiple therapies that are improving the lives of their children. But I'm not here to set blame. I'm here and our community is here to be part of the solution. Today, we find ourselves spending a huge chunk of time worrying about 2 key areas: patient access and therapy approval. They go hand in hand, and to a parent, they are very important. Regulators tell us there was not enough information in a 1-year control study to demonstrate these therapies are working, and the measuring tool we used was no longer sufficient. While we recognize that the FDA has a rigorous approval process, often to a rare disease community, there must be more flexibility for those with dire unmet medical needs. So let's talk about how we can do that together. Let me speak to access first. Even though I know this isn't about access, it's important in the therapy approval process. And then I'll talk about therapy approval. I go back to an analogy of college football. I work at Notre Dame. In college football, there's something called the coaches poll. That is where coaches from around the country rank different teams. But what are coaches actually doing on a Saturday when all the games are being played? They're coaching their own team. They're not watching any of the other teams play, and then they have to make a ranking based on scores of the other games only. There's so much more that happens in each of these games that they are missing and should impact the scoring system. That is what a rare disease community is going through. They're intimately involved in every aspect of this disease. They see past a 1-year placebo-controlled experiment. They do see years of expanded access days showing the effectiveness of a therapy. They see differences in their own children before and after. They listen to a school teacher tell them, you know, in the past 3 months, I noticed a big difference in your daughter. She's been way more attentive, participating in class, playing with her friends. What's the difference? And then the parent says, oh yeah, well, she started a new therapy 3 years ago. When a parent hears this and sees years of EAP data confirming what they're seeing, they will fight to the end to keep access to this therapy, and we will fight with them. I recognize if regulators had the budget to be intimately involved in every disease, they would. And they simply cannot use this type of data for an approval. But we hope they would consider all this when it comes to patient access, which leads to the second item I mentioned, therapy approval. As long as patients have access, we can focus on the extremely difficult challenge to gather enough data in a randomized placebo-controlled trial. We were very impressed by a lot of great ideas from the Duke Margolis workshop. Can we identify one or two specific domains within the NPCCSS or identify a prognostic biomarker? There's a lot to take in and we want to collaborate. So what are we doing? This past year, following the Duke Margolis workshop, we were inspired to launch the NPC Biomarker and Clinical Endpoint Initiative. We are focusing on three key themes: improving the NPCCSS, identifying advanced candidate biomarkers, and identify potential intermediate clinical endpoints. We've raised over $1 million for this initiative and will continue to raise the necessary funds to get these projects over the goal line. In May, we hosted a follow-up workshop with members of the NPC community, industry, the FDA, and the Critical Path Institute. And then we also supported a massive mass spectrometry biomarker discovery program this past year. I don't know all the fine points about biomarker development, but we do know a few things. We are committed to raising funds for this. We do know these three things are critical for the drug approval process and it needs to happen now. And we know we need the FDA as a partner. So I thank the Duke Margolis Center and the FDA for helping us to inspire the launch of this initiative and their participation. I thank all the NPC families and their advocates that fought for access and therapy approval. And I simply have one request. Please, please support patient access to all therapies while we continue to work with you to identify biomarkers and endpoints. Thank you.
Thanks very much for your comments, Sean. And I appreciate your efforts on the NPC biomarker and clinical endpoint initiative. I'd like to turn now to Dr. Denny Porter for a researcher's perspective. Dr. Porter is the clinical director at the National Institute of Childhood Health and Human Development. Dr. Porter, thanks for being with us.
Thank you very much. And I want to start by thanking the participants and organizers of this workshop. It is extremely important that we find a path forward to impact this devastating and lethal disease. We need to work together to find this path. The focus on this workshop was primarily on how we show the efficacy of a single drug within the confines of a short clinical trial. That's important. I don't want to discount that. But we need to be careful not to lose track of the ultimate goal. We need treatments, plural, which impact neurological progression over 5, 10, 15 years. We need treatments that are initiated prior to permanent neurological damage occurring. For the majority of individuals with NPC, disease progression is measured in years, not in months. I believe everybody is in absolute agreement that the NPC neurological severity score, and specifically the 5-domain subscale, can be improved. Although significant data do exist to support the use of this tool to describe disease progression, especially over the course of the disease, evidence gaps do exist, and harmonization of its implementation is absolutely critically important. We appreciate the time and effort by multiple people, by multiple individuals to provide constructive feedback and suggestions. Similarly, although it compounds the issue of rarity, defining more homogeneous subgroups may decrease phenotypic variability. These approaches may help. However, unfortunately, many of us who have studied and understand this disease are concerned that these approaches will not overcome the inherent limitations of trying to obtain accurate and predictive evaluations of therapeutic agents in the confines of a short placebo-controlled trial. Today, we're focused on NPC, but NPC is not the only ultra-rare disease where traditional approaches are becoming an impediment to advancing effective and risk-appropriate therapies. Perhaps this doesn't even get restricted to rare diseases. The concept of personalized medicine will subdivide even common diseases into smaller and smaller cohorts. As a scientist, we apply scientific methods and experimental approaches to answer specific questions. Some advancements are made by applying these methods to different problems and questions. The questions being asked are constructed in order to allow existing approaches to be applied. However, to truly advance science, new approaches and methods are needed. My concern is that we're focused on adapting the disease to the methods when we really need to are methods that are adapted to the inherent issues related to rare diseases. The scientific methods employed need to fit the problem, not vice versa. We have a clear example of a safe and effective therapy in NPC. A decade ago, I had reasonable doubt that miglustat was effective, but it also was reasonably likely, and by luck for some of our kids, it was available off-label if one could persuade an insurance company to cover the costs. I joke about how our work was actually randomized by insurance companies, but that's actually a sad joke. These kids and families deserve better. The EMA approved miglustat, as we all know, based on positive trends, definitely not strong evidence, but they mandated monitoring. This allowed for collection of real-life, long-term data to demonstrate efficacy. Today, I don't know a physician who deals with NPC that does not consider miglustat to be standard of care for NPC, individuals with NPC. In fact, queries from the agency have actually asked for justification to discontinue miglustat therapy. The real-life data is unambiguous. Miglustat slows disease progression and extends lifespan. The key is that there was time to collect the data. There was time to apply an approach that fits the problem. Today, I have to ask, what scientific or clinical knowledge would now accrue by having a placebo-controlled trial? How would that advance our understanding of how to treat this disease? And how would it help our patients? It is simply trying to adapt the disease to a method. Short-term randomized clinical trials are neither a rational nor feasible approach to achieve our ultimate goal. We need acceptance of new models that allow for long-term study of clinical impacts that incorporate the concept of totality of data, including natural history data and including individual patients' trajectories. Yes, it's messy data, but we have to use it. We need acceptance of models that recognize the value of biomarkers that are directly and proximally related to NPC1 pathology. Their value may not meet the standards as a surrogate for a clinical outcome, but they have significant value if the standard of reasonably likely is applied. We need the agency to exercise the regulatory discretion of reasonably likely that is the legislative standard for accelerated approval. Personally, I think the European term of conditional approval is better. No one is looking for a quick way to cut corners on establishing a safe and effective therapy. On the contrary, the NPC research community is looking for the time that will be necessary to truly establish in a scientifically rigorous manner that a drug is effective in slowing the progression of this devastating disease. Again, this is a disease that manifests over years, not months. Reasonably likely also does not mean that there should not be reasonable doubt. In fact, there should be. A standard of reasonably likely, if applied appropriately, also means there will be failures. As a researcher, if all my experiments gave positive results, I'm not asking questions that will move the field forward. Both regulatory agencies and the NPC patient community will need to accept this. The NPC community will also need to understand that longer and clinical trials will not be easy or quick. However, if we focus on defining a path forward that recognizes the inherent aspects of the disease, takes advantage of a strong understanding of both the pathology and natural history of NPC, and provides time to clearly and rigorously demonstrate which therapeutics interventions are truly effective in slowing this disease, we will have the opportunity to make a real and significant impact for individuals with NPC. If this is our goal, it needs to be a goal where we work together to achieve this, and it's achievable if we work together.
Thanks very much, Dr. Kaysen, for covering a lot of ground on the clinical research side. I'm sorry, Dr. Porter, for covering so much ground on the clinical research side. And I'd like to turn now to Dr. Jacqueline Karp for a presentation on the January workshop, key themes and future directions. Dr. Karp is the clinical team lead in the Division of Rare Diseases and Medical Genetics in the FDA's Office of Rare Diseases, Pediatrics, Urology, and Reproductive Medicine. Dr. Karp, thanks for being with us today, and thanks for the presentation.
Thank you, Dr. McClellan. Can you hear me?
Yes, just fine.
Good afternoon, everyone. I would also like to thank everyone here for their participation in this webinar today and for all of your efforts to support development of therapies for NPC. Next slide would be great, please. Thank you. So again, at the January workshop, participants discussed the challenges and opportunities in NPC product development with a focus on endpoints in clinical trials. In this talk, I will give a recap of each of the 6 sessions from this workshop, which are listed here. Next slide, please. So most sessions started with a perspective from a patient or caregiver, indeed, the most important of stakeholders here. In session 1, a parent of 2 NPC patients emphasized that although NPC is considered a slowly progressive disease, from the perspective of patients and families, it isn't slow when you are told you only have a matter of several years left. I think that's a very important point to keep in mind as we consider endpoints and optimal ways to support product development. It really captures the urgency of the need to develop and identify effective therapies for this community. As a reminder, the 5-domain NPC Clinical Severity Scale was created to support research by using the 5 functional domains selected as the most clinically meaningful to patients, caregivers, and clinical experts. And these domains are listed here. While the scale has been a resource for understanding disease progression, there have been some challenges with interpreting its results, especially in short-duration clinical trials. For example, there are some limitations in its validity evidence, and that is evidence that it is actually measuring exactly what it intends to measure that makes score interpretation difficult. The cognition domain is especially challenging. Although cognition is critical for long-term assessment of patients with NPC, evidence from the natural history study indicates that declines occur over many years, and therefore assessments may not detect change within the time frame of a typical clinical trial. Next slide, please. Opportunities for addressing these limitations were discussed, including the need to generate evidence that the different score options within a domain do not overlap, are consistently interpreted by clinical experts, and are relevant for the full age spectrum being studied. There is also a need to ensure that the scale has standardized implementation, that the same assessment happens the same way with every patient by every clinician at every site. Strategies here could include harmonized training materials and standardizing all assessments. There was also discussion about how interpretation of clinical trial measurements could be enhanced with the use of new methods such as biomarkers. Next slide, please. Start off session 2, another parent of 2 NPC patients who is also a speech pathologist highlighted the need to include patient and caregiver observations as part of swallowing evaluation protocols. Swallowing difficulties are a key symptom of NPC, with more than 80% of patients developing them, so it is critical to determine optimal ways to measure swallowing. Assessing swallowing as a clinical trial endpoint can be very difficult. Challenges in the interpretation of these studies stem from the heterogeneity of the disease and lack of standardization in assessment methodology. The video fluoroscopic swallowing study, one evaluation often used, is considered the standard for adults, but can be challenging for younger patients to comply with. Another option is the fiber optic endoscopic evaluation of swallow, which can be very difficult for many patients to tolerate. And a limitation of both of these types of studies is that many clinical sites are not equipped to perform them, given the specialized training needed for their administration and interpretation. There are other burdens with swallow studies that can make participation difficult and the measures less reflective of daily swallowing abilities. For example, a patient may struggle only with certain food textures that are not used in the assessments, or some patients may have difficulty tolerating the taste of foods that are used in these assessments. Additional burdens include the long distances often required to travel to sites equipped to perform the studies. This may decrease the population available to participate in trials or introduce bias. Next slide please. On this slide are listed some approaches participants highlighted that may help address the limitations of swallowing measures. Patient and caregiver diaries may help communication and fill in gaps between assessments performed during visits. Additionally, newer technologies may make swallowing evaluations more consistent by allowing videos taken in multiple locations to have the same evaluator. Additionally, planning discussions can be beneficial for evaluators to have with patients in advance of the assessment. It was also noted that overall, the speech-language pathology field is becoming more standardized, which may lead to additional opportunities for improving accessibility and standardization of swallowing measures, which would reduce the limitations for their use in clinical trials. Next slide, please. At the start of session 3, another caregiver expressed how critical it is to recognize not just a stopping of disease progression, but also a slowing of progression as a massive success. Ambulation, speech, and fine motor skills are other outcomes identified as most meaningful in NPC. As with swallowing, functional measures of these outcomes also have several challenges. Often, these measures are categorical scales that may not be sufficiently sensitive to detect meaningful changes. Stabilization or slowed progression may not be captured, even if meaningful to patients and caregivers. Additionally, standardized scales may not accurately reflect how a patient functions in the real world. Also, since these measures rely on episodic measurements, they could miss key events or mischaracterize trends. All of these limitations could potentially lead to false negative results in clinical trials. Functional areas can also be very difficult to assess for patients across the age spectrum. For example, very young children would likely have not reached some developmental milestones relevant to selected assessments. Another limitation of these functional measures is that they can be subjective and heavily dependent on a patient's level of engagement with the administrator. Next slide, please. Okay. Important strategies for addressing the limitations of these functional measures include determining how multi-system impairments may impact assessments. For example, how cognitive issues can impact performance on fine motor assessments, and ensuring the assessments are adequately sensitive. This could be accomplished by incorporating multiple assessments and multiple types of statistical analyses to optimize these measurements, which could include use of raw scores, normative scores, and age equivalents. Next slide, please. At the start of session 4, a caregiver stressed that while patients and families are sharing the meaningful outcomes they see in their day-to-day lives with investigational therapies, they feel that information is not necessarily being adequately captured or considered. a potential opportunity for overcoming this particular challenge and some of the other challenges with functional measures I just described is the use of digital health technologies, which may offer additional opportunities to collect meaningful and more frequent data. Through the use of telehealth, these technologies could also expand access to clinical trials by requiring less travel and facilitating assessments in a home setting, which may better capture the full range of a patient's abilities rather than just a snapshot. Furthermore, videographic analysis could increase inter-rater reliability by having a consistent reader. There are limitations of digital health technologies that must be considered, such as the ones listed here. Participants seem to agree that the best initial use for digital health technologies in clinical trials would be in a complementary role to traditional clinical data collection rather than as a replacement. Next slide. The development of biomarkers has the potential to support and enhance development by assisting in diagnosis and in assessing disease progression and response to treatment. Additionally, compared to other methods, biomarker testing could be faster, lower cost, and less invasive. However, to be used in regulatory submissions, biomarkers require analytical and clinical validation. A biomarker that has been validated to predict a clinical benefit could be used as a surrogate endpoint for future trials. Robust biomarker data could also be used as confirmatory evidence of effectiveness alongside data from clinical assessments. Next slide, please. Research has already identified several diagnostic and potential prognostic biomarkers, such as the ones listed here. FDA recommends measuring these as exploratory endpoints in all trials. Accumulating evidence may demonstrate that one or more of these can work as endpoints to speed development and approval of drugs. There are limitations with the use of biomarkers, such as the ones listed here. It is also difficult to statistically correlate changes in biomarkers to clinical improvement based on data from very small populations. And answering these unknowns, or clinically validating the biomarker, is again needed for its use as a surrogate endpoint. Because many of the biomarkers under investigation are relevant to other diseases, there may be opportunities for collaboration among research in other disease spaces. For example, neurofilament light chain listed here has been explored for many neurologic diseases and is a candidate for assessing neurodegeneration. So overall, there have been significant advancements in the understanding of biomarkers for NPC, but significant challenges remain. Research to move this area forward could provide major advances for NPC therapeutics in the future. Next slide. At the beginning of this last session, a caregiver reiterated how critical it is for patients and families to have a voice in identifying meaningful clinical outcomes. Participants emphasized the importance of remembering that because therapies may not result in improvement in disease symptoms, the goal is to understand whether a therapy may result in stabilization or attenuation of the decline in the rate of disease progression. It is critical that endpoint development and selection reflect this goal. Another caregiver during this session also emphasized how researchers and regulators cannot lose sight of the full spectrum of patients impacted by NPC, including adult patients, and the differences in their needs and priorities. Next slide, please. So getting into some specific strategies in pursuit of these goals, participants stress that improvements and modifications to the 5-domain NPC clinical severity scale must be able to integrate with the existing data. And given the widespread use of investigational and off-label products, the existing natural history data may never be replicated. Participants discuss the importance of bolstering the available data rather than recreating this data set to best support development. Participants also discussed a number of potential strategies for making the development process more efficient, including sharing data, continuing engagement between regulators and patients and caregivers, and the other strategies listed here. Next slide, please. And the last one. So a key recurring theme from the January workshop was the importance of collaboration in supporting therapeutic development. As you heard Dr. Cavazzoni mention, the FDA has several initiatives that will help facilitate collaboration, such as the ARC program and work with the Critical Path Institute. As we move forward, it will be very important to continue and build on these efforts. In closing, I'd like to thank you all again for your participation today and for the critical work you do for the NPC community.
Great. Thank you very much, Dr. Karp, for that presentation. You covered a lot of ground from the workshop in just a limited period of time. We do have some time now for questions and comments. We've had a couple in already. We're also have some that came in before. I'd like to start with one of those, and I guess let me first introduce our Q&A respondents. So a number of them you've met already or heard from already during this session. Dr. Karp, Dr. Kaysen, Dr. Porter, Dr. Hopkin. Also want to introduce Katie Donahue and Naomi Noble, who are joining us from the U.S. Food and Drug Administration as well. Thanks to all of you for being with us and for helping with this part of our webinar. The question I'd like to start with, maybe Katie, if I could direct this to you and bring you in. We just heard from Jacqueline with a nice summary about the insights shared during the January workshop, and many have here today referenced the summary report that followed the workshop as well. Could you comment a bit on how the workshop and the report are impacting FDA's approach to NPC drug development? That's come up in a number of the comments and questions that we've received already.
Thanks, Mark. Yeah, I mean, I think, you know, the core takeaway from this community in the endpoint session and in other interactions that we've had is they're telling us that the scientific measures we have are failing to detect benefits that matter to patients within a reasonable amount of time. So the endpoint we have doesn't measure things that matter to patients soon enough for folks to be willing to enroll in placebo-controlled trials, right? And the challenge is that with this clinical outcome assessment, the NPCCSS, you have to have a placebo arm if you're going to use that endpoint. And it sounds like you're going to have a placebo arm for longer trials, you know, 2, 3 years to generate evidence on that endpoint. And obviously, this community is very clear that they're frustrated about those things. And so I'm so excited to hear Sean's news and the Persegian Foundation's news that they're investing in biomarker development. So to me, that is one of the most exciting developments for this community. And it's important because it would address a lot of the challenges that we're hearing right now. Now, it's probably not a panacea, and I have no doubts that the community is going to think they've cleared the evidentiary bar for the biomarker before the FDA agrees. We're probably going to still have some disagreements there. But I'm excited to see the work developing because I think it's really critical. You know, it can be stepwise, right? So things that we can leverage some of the existing natural history data. I mean, any patient who has a sample, like a serum sample stored in a research freezer somewhere, whether from a clinical trial or an NIH study or whatever it is, well, those can be analyzed for biomarkers and the data correlated to their NPCCSS scores and other clinical outcomes. And we can start to build that evidentiary basis. And so to me, that's what I think needs to happen. And those data need to be shared with some sort of central repository. So having little bits of data here and there and everywhere isn't going to get us where we need to go. It needs to all be shared to a common place like the RDCA DAP or something similar with CPATH. And so I think I'm excited to see the community sort of coalescing around that work, because what would a biomarker do? Well, first, it would allow for some dose ranging. So one of the things I've heard is the community is so frustrated. They've had 4 well-done randomized clinical trials that have failed. And this is a disaster. Like, we can't keep doing the same thing over and over again. Yeah. And one of the big missing pieces has been dose ranging. And one of the problems is the community doesn't have great biomarkers for dose ranging. And so advancing biomarker development means we can do dose ranging in a reasonable amount of time and maybe have a better shot at picking the right dose to carry forward into a successful trial. And then with more evidence around the biomarker for dose ranging and from all of these other sources, natural history and other, completed clinical trials, well, if we get a biomarker that is reasonably likely to predict clinical outcomes, and Denny, I want to touch back on that reasonably likely standard is for accelerated approval for biomarkers. The lawyers won't let me apply that standard to clinical outcomes like the NPCCSS. The law says no. So we got to have a biomarker in order to consider the accelerated approval pathway. And once you have really strong evidence that the biomarker is predicting outcomes, we could probably get away with single arm trials. So as we get more and more and more evidence, we can, for a biomarker, that is going to solve a lot of the challenges that this community is grappling with now. We're not going to get there quickly. It's not going to be easy, but I think it's hopeful. So.
Thanks. And just to follow up on that, since several of the questions that have come in touched on the issue, as you did, on how natural history data can best be incorporated and potentially get to the point where you have single arm trials, I guess, is a goal there too. You talked about that in your response just now. So getting those data into a standardized format, building out a consistent evidence base, the kinds of steps that have been undertaken since the workshop. Is that the right direction there? Any additional comments about how natural history data can be used and maybe reduce the need for placebo-controlled trials?
Right. I mean, having a biomarker that we know, so that's no longer reasonably likely, but we're down the road a ways, and we are now convinced that this biomarker predicts clinical benefit, much like the evidence we have, for example, for cholesterol and heart disease, right? So if a new drug treats cholesterol, they don't get accelerated approval, they get full clinical approval. And so we might be able to see, and some of this is going to depend on the variability in the biomarker and the measurement properties and that sort of thing that we still have to explore. But a lot of cancer trials are single arm trials that measure tumor size on CT scan. So if we get a biomarker that we can have a similar amount of confidence in, then maybe we could do a similar single arm trial where we're seeing that this biomarker never goes down unless the patient is in fact getting better. If we see a lot of noise in the biomarker, we may still end up needing a placebo control, but some of it's going to depend on how those biomarkers function, what happens in NPC patients. So that's what we have to explore and understand in order to kind of move it forward.
Great, thank you. Dr. Porter, I know you talked a lot about the use of biomarkers and validation of biomarkers and getting to that reasonably likely, meeting that reasonably likely standard as Katie was just describing. Would you mind commenting a little bit more about your thoughts on how to get to biomarker validation as quickly and efficiently as possible?
So, and just to clarify, reasonably likely was stated in the context of thinking about biomarkers. I may not have been clear.
Right.
We have candidates. We have candidates that clearly relate to proximal aspects of the disease. We have candidates that relate to the pathology of the disease. If you put a standard that something never happens, that's not reasonably likely. We know that from clinical medicine, and we know that from the variability in this disease. There will be variability in the biomarkers. But what we need to do, and I think what you were outlining, is what is their association with the disease progression? They actually do associate with the disease progression as measured by the neurological severity score. So we have to come to a point where we recognize how we can assess clinical progression, and clinical progression over years is accepted. And then I think we have to come to a point where the associations, there are associations, it's not guaranteed, it's a probability, that those associations meet the standard of reasonably likely to predict that we are impacting the pathology of the disease When we impact the pathology of the disease, that is reasonably likely to lead to clinical improvement.
Great. Thank you. And I'm sorry, go ahead.
It'll take years to show the clinical improvement. We need time to show that.
Thank you for those comments. And we have just a few minutes left. I wanted to go on this important issue of biomarker validation back to Dr. Karp. Any comments you'd like to add to Denny's remarks about validation?
I mean, I would just reemphasize what I mentioned in my talk. I think one of the key things the community can do to advance the development of biomarkers as potential surrogates is to include a robust panel as exploratory endpoints in all NPC clinical trials, and have a plan to share the results to a common repository. Again, going back to the data sharing. This is how other rare disease patient communities have advanced the science for surrogate endpoints to support shorter clinical trials, faster timelines for approvals.
Great, thank you. And we are almost out of time, but I would like to go back. We started out with some comments from the patient community. Maybe Dr. Kaysen, Dr. Hopkin, any brief remarks you'd like to make as we end this session?
I can start, Sean. I again appreciate the ongoing engagement. I think from a community standpoint, it sounds like we have been given direction. I think hearing the idea of long-term clinical trials and development years down the road is not the news that we probably were hoping to hear as a patient community. I think we're hopeful that the clinical severity scale can be modified, it can be used, and it can be really an endpoint in clinical trials on a relatively short amount of time. It's hard to think about proving a biomarker is tracking disease appropriately when we don't have therapies that we're getting approved that can show improvement in disease. And I know the animal models and other things can help with that. We hope to work together to get a path forward so clinical trials in the future can be helpful, but I'm going to circle back and really look at our many programs which clinical trial design that were developed at the time with partnership, I believe, with regulatory that did not have these sort of parameters. And I believe that they're meaningful, and I hope we can get those approved while we're working toward a better solution in the long term.
Great, thanks, Justin and Sean. Brief comment?
Sure, yeah. Again, thanks for everyone's time today. We said that we're investing in, multiple areas, but obviously biomarker development is one of them. And we really look at collaboration on that. We have a great ability to raise resources through our foundation, through families and our army of supporters. We have great scientists, clinicians working on this problem now, as Denny mentioned. But what we do need is help. We need guidance on multiple levels. We'd like to move fast. We're ready to work now. We don't want to wait another year and have another workshop. We need guidance today so we make sure we're moving in the right direction on this. And that's where I really value working with the FDA and other organizations like CPAP and really hope to move quickly on that.
Great. Thank you all very much. I really appreciate the comments and perspectives and putting so much information into a limited amount of time today. We managed to cover a lot of ground in this brief event and highlight both the importance and the value of this collaborative effort to make faster progress to help all of those who are affected by NPC. I do want to conclude with a thanks to the FDA staff who put a lot of time and effort into the workshop and the webinar and this expanding set of collaborative activities that are underway. And also a thanks to the Duke Margolis team, Tori Gimme, Ethan Chupp, Dure Kim, Nancy Ann LaPointe, Luke DeRocher, and Morgan Romine for their efforts today. And again, special thanks to all of you who've come together. I hope you enjoy the rest of this summer day, and we look forward to continuing to work with you for progress on NPC. Thank you very much.